Okay, it's not like there's anything else going on today. Not like we're in the middle of a hotly contentious election. We have more important things to talk about, like cloacal drinking. Sorry, I have to throw in as many cloacal drinking jokes as I can. Okay, so today the topic is going to be Blackhead disease, and I guess I'm actually kind of becoming an expert. Like, we're starting to do some research on Blackhead disease because I started some conversations with some collaborators in the poultry science department, and they told me about this disease, this Blackhead disease, and when I sort of did some preliminary research into it, I told them, you guys had me at cloacal drinking. Sorry, I have to lighten the mood. Okay, so Blackhead disease is let's see, where's my thing? Okay, so Blackhead disease is the name. This is a disease of poultry, of birds. So very important in the poultry industry where they're raising turkeys, and also not quite as important, but also important in chickens. And if you didn't know, Alabama is the #1 producer of chicken in the United States. So this is why we have a gigantic poultry industry and an entire department of poultry science at Auburn. So there's a lot of really interesting research into poultry science, and this is one of the diseases that we are beginning to do some research on. So history of the disease. The disease was discovered in 1893 in Rhode Island. Okay. And it was first discovered in turkeys. It has its most pronounced sort of, I guess, symptoms in turkeys. It has extremely high mortality in turkeys. So you can sometimes lose 100% of your, what do you call it, your flock. Again, I'm not a bird expert. I'm a bug expert. But so if I get some terminology wrong, don't be upset about that. But it's very serious in turkeys, and so it was discovered when it decimated a population of turkeys in New England in 1893. And a few years after that, in subsequent years, there were about a total of 4.4 million turkeys a year that died of Blackhead disease. So it's very, very pronounced in turkeys. And it's actually like if you have these outbreaks, it actually becomes literally kind of infeasible to be a turkey farmer. If there's so many outbreaks, you can't make profit, or you're at huge risk if there's outbreaks of this thing breaking out. So people actually stopped farming turkeys for a little while because of this disease. I would also say, although it was discovered in America and Rhode Island, this is a worldwide issue. It's a worldwide disease. It's all over the world. And poultry is big in every country all over the world. So this is a worldwide issue. And it periodically pops up. So there's periodic outbreaks of it. So where we research it in Alabama is in chickens. Okay. And in chickens, they can die. So sometimes you can get like 10 to 20% mortality, but usually that's not the case. Usually it's just that chickens become what we would call like asymptomatic reservoirs. So they're not going to die, but if you have a farm that is rearing chickens to be sold, or rearing chickens for meat, or potentially having chickens, broiler breeders that lay eggs, this can sort of like decrease the productivity of your agricultural farms. So it is something we care about in chickens, and we do take it seriously. So and I'm learning this as I go, but in the chicken industry, in the poultry industry, so there are what are called broiler breeders. So broilers, I have ascertained, are Broilers are like the chickens you rear from chicks to an older age, and then you slaughter them for meat. Those are what we call broilers. So then broiler, there's something in the chat. Did I get something wrong? Yeah, food chickens, broilers. Good, excellent. If I say anything wrong, correct me. And broiler breeders are then the mama hens that lay the eggs that then are sold to be raised as meat chickens. So not only are there farms of broilers, which produce meat, but there's also farms of the broiler breeders, the hens that produce the eggs. And then the broilers, the broiler farmers buy those eggs or buy those chicks to put them in their farms. So there's different types of farms. And in broiler breeder farms, so these are the hens that are laying the eggs, this can cause a reduction in their egg lay. And so you get a loss of productivity. Okay. So this is a vector-borne disease. But it's also, it has a vector, but it can also be spread bird to bird directly. So let's talk about some of the mechanisms of how this thing spreads. So if you have a chicken, something, something like that, chicken. Chickens, okay, they have an intestinal system, right? And then their anus, I guess, is called the cloaca. I don't know why it's not called the anus. There must be something about birds, whatever.
I think it's a dual function. It doesn't just function as an anus.
Oh, really? Okay. Okay, so it's a multifunctional rectum. Okay. The cloaca, which is right there. And this disease, blackhead disease, can spread through cloacal drinking. And at first, when I heard this, I thought it was something different. I thought it was another chicken drinking from the cloaca of another chicken. That's not what happens. I guess it's actually, it's like a reflexive instinct of the muscle, where when the chicken's cloaca is in contact with like fluids, like maybe it's sitting on the ground and there's some fluids there, the chicken's cloaca reflexively sucks up and absorbs some of like the liquid on the ground. So I guess the first question is like, why would a chicken cloaca ever do this? Could this be beneficial for anything? Clean it out? Man, it's a decent thought, maybe. Suck it in and out and spit it spit it out, maybe. I don't know. How well? Okay, let's see in the chat. Maybe you could drink if they get dehydrated. Somebody suggesting that maybe they drink from it. What I've read, interesting snake scan. What I've read in the literature is that this is a way for them to pick up beneficial microbiota. So like in your guts, right? We have digestive microbiota that help us digest things. Chickens are no different, and it's thought that the cloacal drinking is maybe a mechanism through which they can intake beneficial microbiota from their environment. Okay, so blackhead essentially gets, we'll talk more about the life cycle, but essentially it can get crapped out. So it gets crapped out like in the pathogen will get crapped out in chicken poop. And then obviously if another chicken does some cloacal drinking on the chicken poop, it'll get the blackhead disease. So so that's one mechanism through which it can spread. Okay. I know, that's why I said you had me at cloacal drinking. Like when I was offered to start researching this, I was like, this is totally cool. Yeah, it's right up my alley. Okay. Let's talk about we'll come back to this because there's more mechanisms through which it's transmitted, because it seems to be a vector-borne disease, but it's complex and it's bizarre. Okay, so let's talk more. So the pathogen of blackhead disease is Histimonus Meleagritis. Okay, and I pulled up the Wikipedia. I was hoping there'd be like a picture of it. Let me see if I can just quick Google Histimonus. Let me see if I can just quick find a picture of it. Because it's like an amoeba. Okay, here's some pictures. Oh no, these are worms. It's very strange. It replicates by binary fission. I'll just draw kind of maybe what it would look like. So it has two different forms. One form is what we call an amoeboid form. So it's a protozoan, it's a eukaryote. Okay, it's not a bacteria. And amoeboid, we should know amoebas are these creatures that they have pseudopodia, which means false feet. They crawl by sort of organizing actin and cytoskeletal polymerization on one end of their cell, and then that forms what's called the pseudopod, the false feet, and then they just sort of like move things there, and that's how they grow. So we should kind of know what amoeboid means. So one form of it is amoeboid. When it's inside the chicken, it becomes what we call the flagellated form. And perhaps it won't be accurate as I draw this, but I've read that they have 3 flagellas. So in this form, the flagella would be used to sort of like swim or navigate through tissues. So it has different forms. Not only that, the Histimonus, it's not obligate, but it doesn't survive well outside of a host. So it's not, I would say it's not environmental. So like, it's not like it's not like you're gonna pick up a patch of dirt and find living Histimonus meleagritis in there. It mostly survives in what we would call the reservoirs, or in some cases, they are called the paratenic hosts. Okay, so now is where it starts to get even weirder. So it's a vector-borne disease, but it's recursive vectors. There's vectors inside of other vectors. It gets very strange. Okay, so there is another organism that's important for this called Heteracus Galinarum. This is the chicken cecal worm. Okay, what is the cecum?
I think
Yes, very good, excellent, perfect anatomy. So in the chicken, in the transition, just as was mentioned, in the transition between the small intestine and the large intestine, it's like a little pouch or something like that. I guess maybe kind of like our appendix, something like that. That's called the cecum. And there are nematodes, so tiny filarial worms, nematodes, that infect the chicken cecum, and they're called the chicken cecal worms. Their species is Heteracus galinarum. So these nematodes are where the Histimonus lives. And every time one of these things molts, it releases some of the some of the Histimonus into the chicken. Okay, so the reservoir is not only is the reservoir chickens. That's kind of like, okay, I guess that's actually kind of like the secondary reservoir. The primary reservoir is the cecal worm. Okay, and these cecal worms, like any nematodes, they lay eggs. And when they lay eggs, the Histimonus is maternally passed on. Not at high efficiency, but at about 25% is what I've read, efficiency of maternal transmission. So when these nematodes lay eggs, they're laying them with a decent sized infection rate of Heteracus. And they can lay eggs that just kind of sit for a while. And they can lay dormant. Okay, and these eggs can survive in the environment, and the worms can survive in the chickens, and all this gets pooped out in the chicken crap. Okay. So let's draw out more of the complexity of this life cycle. Let me just say a couple quick things about Histimonus. Now, see, here's where things get really confusing. When you first start to introduce this, you get Heteracus and Histimonus confused all the time. So Histimonus is the pathogen. A couple other things about it. It's really strange. There's no mitochondria. No mitochondria. And what was the other thing? Oh, it's amoeba. It's an amoeba, and amoebas are predatory. Amoebas are like predatory creatures. They go and they actively hunt out things and they eat them. And so Histimonus actually eats other bacteria that are in the system. So the strange thing is, right, like chickens have their microbiota. In chickens, you often find E. coli, Bacillus subtilis, and this coccidiosis thing, this coccidia thing. And this amoeba eats these as like food. So sometimes when blackhead gets real bad is also when there's big co-infections with perhaps not necessarily pathogenic E. coli, but when there's co-infections with other bacteria that the Histimonus can prey upon. Okay, so it often is said to get worse when there's lots of other bacteria present in the system, because it can eat those and then get energy and then replicate. So that's another sort of unique, interesting thing about this system. Okay, now let's draw out more of the complex life cycle. Okay, so if you're the chicken, you have the, let me get a different color. You have the cecal worms, they'll be red, cecal worms inside of you. And they're pooping out these cecal worms, and the cecal worms are laying eggs in the crab. Okay, so one mechanism of transmission would then be the cloacal drinking. Okay. Another mechanism you could imagine, I don't know, I guess it probably unlikely, but there might be certain scenarios where chickens end up taking in some of the crap directly into their mouth, perhaps, although unlikely. But there are some scenarios where that happens through an indirect route. So now imagine you're in a poultry farm and there's lots of chickens and lots of chicken crap. There are lots of things that love to eat animal poop, okay? So one of these is called, what we call common name, litter beetles or darkling beetles. They're little black beetles. And if you go into a chicken coop or a farm, you find these like in the millions, because they have like an abundant food source where they're living in and eating the chicken crap. And the chickens will then eat the litter beetles. Okay. So another thought is that you have these insects, and if these insects are living in and consuming the chicken crap, they're also taking in the Heteracus worms and the eggs. And if a different chicken eats this, okay, it's gonna now get that egg into its system, and now that is gonna reinfect other chickens with not only just the cecal worms. So the cecal worms themselves are a vector-borne disease, potentially vectored by transmitted by things like litter beetles. But also, if those cecal worms are infected with Histimonus, then they're taken along with them, right? And it's not just litter beetles. So people in the lab have demonstrated that there are other insects that can pick up these eggs and, in theory, mechanically transmit them. So there are blowflies. Blowflies are like calphorids, which are like the metallic green blowflies you'll see if like dog pooper in your house. Those are blowflies, and they're attracted to feces, so they'll go in there, they'll get infected. There's a paper that suggests grasshoppers, although I've looked in the chicken barns, you don't see grasshoppers very much. We didn't find any when we looked. Blowflies for sure, you'll see them, and darkling beetles are the most abundant thing. And then there's other people that have reported earthworms. So the thing about these chicken barns is they don't have a concrete floor. Okay, so what it is it's a giant barn, and it's pretty gigantic. Like you walk in, you're about this size, and there's a door on one end and a door on the other side, and there's dirt, and over the dirt they put what's called the litter, and there's a bunch of chickens in there, but the earthworms can directly like come in contact and interact with the litter. Okay, so another paratenic host are earthworms, which are consuming in theory some of this poop, and if they get eaten by chickens, then they can act as a secondary vector. Okay.
Don't they, like, you know, I think worms, like, go to the liver or something?
The worms go to the liver. That's a good point. So, but not the earthworms. The the Histimonus. It's the Histimonus, not the worms. I actually have a picture of that. Thank you for reminding me. One of the ways that they diagnose this, if you look here, is on Wikipedia. One of the ways that they diagnose this is when the blackhead disease gets real bad, the Histimonus goes into the liver and it causes these like ulcers, these like, what are they called? I don't know what you'd call them. Lesions is what they call them. They call it the lesions. So they cause horrible lesions in the liver, and this is terminal for the turkey, or if it's in the case of the chicken where it does it, can be real bad. So that's one of the ways that they diagnose it is you just take a chicken or a turkey, you cut it open, you look at its liver, and if its liver has these lesions on it, then you start to worry about blackhead disease. So it's very bizarre.
You think you can find Histimonus and liver that you can purchase at the market?
Oh, and like chicken liver?
That'd be cool.
We found two positive PCRs of Histimonus here. I don't know. That's a really good question. I don't know. I mean, I would think that the rate would be I don't know, because actually the rate is actually supposed to be real high. Like actually, like lots of chickens have this.
Because I feel like
Yeah.
people that process chicken aren't trained to look at livers like that.
Yeah, they might. Yeah.
I'll take it. The question is how many people actually consume
Chicken liver. I think actually that's really popular. Actually, like chicken. I've actually like heard of that. It's a good question. I'll ask the experts on that. Chicken liver.
I'll ask that.
Yeah, he'd be a good one to ask. I'll ask Rudiger. See what he says. Okay, so just quick to finish up, and then we'll just go into just a little bit of research I've been doing on this. The way that you can detect this, so detection, is there's a change in the feces color, so it becomes a sulfur color, sulfur colored feces. The liver lesions, which we talked about, you get engorged ceca, so inflamed and engorged ceca. And then you can do microscopy, and if you want to look for the Histimonus, microscopy. And then you can do PCR to test for any of these things. Now, treatment. It used to be not so bad because we had an effective anti-Histimonus drug, which was called nitroimidazole. But that has since been banned. And I would imagine the drugs that you can feed things that people eat is probably pretty restricted. Yeah. So this drug has been banned for use, and so that effectively there's now no mechanism to control for this disease. There's no mechanism to treat for this, and there's no immunity. There's no vaccine. So it is an issue in a sense. We don't really have a way to deal with it. So that's one of the things I'm starting to think about. Like, how could we get rid of this permanently? But in situations like this, in situations where there's no drugs and there's no vaccine, this is an opportunity for us, because what's the control strategy then? What should the control strategy be? Vector control. So that's the case of, so vector control. This is an opportunity for us. This is the same case of dengue, right? Like dengue, there's no drugs. There's really, well, I mean, there's antiviral drugs, but if you get dengue hemorrhagic fever, there's probably not really many drugs that the doctor can give you that are going to help you out. And there's also a vaccine as problematic in that sense, because there's different serotypes, and we talked about the antibody dependent enhancement. So in that case, that was a prime case where vector control is a good strategy. Here it's the same thing. So a lot of the control efforts focus on like deworming, getting rid of the worms, and then also to potentially what we're starting to think about is like if we can decrease the beetle populations and get rid of the beetles, would that help out a little bit? That's kind of what we want to try. Yeah.
I thought nematodes have like a ecological function.
Nematodes have an ecological function.
Like there's like a plus side to them. Like there's a plus side to like having like.
Are you talking about earthworms or nematodes? Earthworms you're talking about? Yeah, earthworms are important for aerating the soil. We're not talking about getting rid of earthworms. We're talking about getting rid of the nematodes. So when I say worm, that's a bad thing. It's kind of like when I say bug. Like bug could mean colloquially like a microbiological thing, or it could mean like an actual hemipteran. When I say worm, I'm talking about nematodes, the ceco worms. But they're actually not, they're not the earthworms, although earthworms are a part of this equation, which is why, again, it's like it's so bizarre and confusing. There's so many different things. So let's I can just kind of like, we have some preliminary data, I guess I could talk about, of like what we did. So this was the Auburn University poultry farm facility, I guess you'd call it. And there was a really interesting opportunity that just came up over the last 6 months, because Auburn University's poultry science department got a completely new facility. So they rebuilt a totally new facility, totally new farms at different locations. So these farms were being like phased out. And so there was a really unique opportunity that many of these farms had not had birds in them for 9 months to 3 years. So we thought, well, let's just go and like, let's just take a look in these old barns that haven't had birds for a while, and let's see what we find. So we went and looked in these barns, and what they call the cake, that's like the shit stacked on top of the shit that's like hardened from the birds. They call that the cake. So we went and we looked at the cake, and there was like 3-year-old cake. And one of the first things we noticed was there's still like robust beetle populations still consuming that cake like years after the birds have gone. Yeah, I want to say 3. I mean, I have to fact check that, but I know for a fact at least 9 months, and I think the highest range was 3 years. I have to double check, but I think the highest range that we checked was they hadn't been there for 3 years. And so the cake is just sitting there, and it's like a foot deep. It's like a foot deep. And so imagine as a beetle, like that's a robust source. So we started collecting these beetles. So we would collect the beetles, and we would collect live ones and dead ones for different barns. And then we just do a simple DNA extraction. And then the first thing we wanted to do is just run PCRs to see if we can detect the cecal worms. So our first screening, that's these data. So it's a little bit zoomed out, but these PCRs are PCRs for nematodes. So this barn was negative. This was M1, which was here. This one was negative. This one was M3, which was positive. So we're getting lots of PCR positive samples in both the live beetles and dead beetles. So one interesting thing is beetle corpses. So they don't even have to be alive. Beetle corpses, dead bodies of old beetles, we're detecting nematodes. Or in theory, DNA from, to be accurate, DNA from nematodes. We don't quite know if they're infectious or if they're alive. But we're detecting DNA from nematodes in these beetle corpses from these emptied farms. This one was negative, M4. That's this one. This one is a little bit positive. Okay, so there's a couple conclusions. There's like a couple things that are worth discussing. The first is that the beetles and the beetle corpses can still hold on to the cecal worms many, many, many months and years after the chickens have gone. So the first thing that this suggests is it might be really difficult to get rid of this disease like once you have it. Once you have the cecal worms, it might be really difficult to get rid of them, because how are you going to get rid of every last beetle, and not only every live beetle, also all the dead beetle corpses would have to clean out. So that would be a really thing, is it's going to be, we think it's going to be hard to like clean this up. The second thing that we noticed was, if the hypothesis, I mean, we're exploring this, if the hypothesis is that the beetle is a vector of the worm, and the worm is a vector of the Histimonus, one of the things we're wondering about is like, well, how good of a vector is the beetle? Vectors that are really good, they fly, like mosquitoes. They fly around, okay? Or they travel, they move a lot. Beetles have wings. These beetles have wings, they can fly. But what we noticed from the data was there were uninfected barns right next to infected barns. So that suggested that beetles were not moving so much because they're not walking 100 feet to the next barn. Does that make sense? Like if they were traveling at least 100 feet consistently, you would expect that all the barns would be positive, right? But that's not what we saw. We saw that just some of the barns are positive, some of the barns are negative. So it suggests the second conclusion is beetles probably aren't great vectors, and they probably don't move much. And it kind of makes sense, like if you have like a lifetime supply of chicken shit, like why bother moving, right? Like you might as well just stay there. So I think that kind of makes sense. And so now we're kind of wondering like, well, what are the conditions under which the beetles will move? And it might be that if you set up some fresh chickens, perhaps maybe the fresh feces would attract more beetles. So that's kind of one of the things we're thinking about. Like how do beetles colonize new barns? Okay, so more into this data. So we did a time analysis where we just went back periodically month after month after month to see if we could, every time we collected beetles, to see if we could detect the nematodes in the beetles. So here we see some detected in June. Here's one detected in a live beetle in July. In August, we were negative, but we saw some in the dead beetles here. And then in September, we saw them. So we could consistently see them at least for the 4 months that we were sampling. So again, persistence is the theme. They persist. The cecal worms. This isn't what we're talking about right now. And then what we started doing is then we started checking not only the beetles, but the litter. So we would go and we would just collect a sample of the litter, do the DNA extraction on the chicken litter, and then run the PCR with the chicken litter. And we did this because then we started to wonder about the beetle movement. So then we started doing this for each pen in this positive house. Okay. So again, the conclusion is the same. In the litter, we were actually able to detect a lot of the Histimonus. It seemed the PCRs were a lot easier and more positive in the actual litter, the feces, than they were in the beetles. But one of the interesting things we saw is that not all the pens were positive. Only some of the pens were positive, and they seem to cluster in this little quadrant. So again, that suggests beetles are probably not moving very much because they're not even bothering to move like 5 feet to the next pen. And so one of the ways, the final immediate sort of like conclusion, what would be sort of a control mechanism that you might suggest if you had seen this data? Well, yeah, I think that's the right idea, but perhaps the wrong word. Like aliquoting the chickens into smaller compartments would be beneficial. So what they do currently is sometimes they have a giant pen, okay, where all the things are just mixing together. If you really want to sort of prevent the spread, it seems like all you have to do is set up individual pens, and then you sort of restrict the movement of the beetles, which are already not moving very much. And so maybe you don't even have to do very much in terms of, it wouldn't actually be that hard. Like it could actually be pretty easy to just do this. And then you would prevent the spread, or you would slow the spread of the nematodes. And then by that, you would slow the spread of Histimonus. So these are just some interesting projects that we're working on. The data is not perfect. We're still working on it. It's actually very difficult to get good DNA extractions from dead beetle corpses and 3-year-old litter samples. It's pretty hard, but the data is interesting, and I think it's gonna lead to a really interesting publication. So that's the end. Any questions?
Is there any regulation when you buy like chickens from like broiler breeders, or do they like test chickens?
Do they test? I'm sure that they do, but I'm not the expert on that. That's another question I should ask Rudiger. Like, what's the testing look like? The other thing that's interesting, I guess that leads me to the other thing. They for sure test for salmonella, because salmonella is a big deal in the chicken industry. And that's the other interesting thing about these beetles, is because these beetles are eating the chicken crap, if there's a salmonella or a E. coli or something like that, or the coccidiosis, they would in theory also be eating those. And so in many papers, people suggest that the beetles might also be essentially spreading everything that's nasty that comes from the chicken, like could be transmitted by these beetles. And so again, it sort of harps on the importance of like, what are the conditions under which these beetles move and how far do they move? That sort of determines how good of a vector they would be. So it's one of the things that we are looking into and trying to understand a little bit more. Good. All right, everybody have a good day.